All right, thank you very much, Marty, for the introduction, and thank you, Jenny and Wendy, for inviting me to speak here. Actually, I will be speaking about a collaborative project that is actually still ongoing between KeyGene and Pacific Biosciences, centered around isoseq transcriptome sequencing in tetraploid cotton, while simultaneously at KeyGene we are engaging in constructing a de novo genome assembly of the tetraploid cotton species. So at KeyGene our passion is to explore and exploit DNA variation in crops, and we like to refer to them as the 6F crops: food, feed, fiber, fuel, flowers and fun. We already heard about the fun part in an earlier presentation, so today I will be talking about fiber crops, but with some side steps to food and feed, as you will observe. So, as another introduction to our activities at KeyGene, we have been working with next generation sequencing technologies for the past 10 years, since the commercial introduction of the first platforms in 2004-2005. And our mission is really to develop innovative applications that advance crop breeding for these 6F crops. And actually today I will be covering a part of what we are doing at KeyGene, namely those applications listed at the left-hand side of this slide: de novo genome sequencing, targeted resequencing, of course transcriptome profiling with isoseq, and also whole genome profiling shown at the right-hand bottom of this slide. So now, as to the outline of my presentation, the objective of this project is really to approach the assembly of a catalog of tetraploid cotton in an integrated way, comprising both genome sequencing and transcriptome sequencing. I will be talking about isoseq and the de novo assembly, as I mentioned. The integration of this genome assembly with a whole genome profiling physical map, which is actually the tool that we have developed earlier for short read platforms, and is very useful to construct both the physical map but also to super scaffold contigs coming out of de novo genome assemblies. And finally, we have developed a targeted resequencing technology that's also useful for gap filling. Finally, we integrate all these sequence data sets as well as other omics data sets in crop-specific encyclopedias that we refer to as croppedias, and in this case that will of course be the Cottonpedia. And then I'll be wrapping up. So to start off with some information on cotton, the Gossypium genus. It's actually a very broad genus of plants comprising 46 diploid species and 5 allotetraploid species. And all these diploid species are thought to have evolved from a common ancestor, diversifying into 8 subgenomes which are designated A to G and K. And all the tetraploid cotton species came from interspecific hybridization between A genome species, Gossypium arboreum, and the D genome species, Gossypium raymondi. So the tetraploid Gossypium hirsutum that I will be talking about today represents over 95% of the annual cotton crop production worldwide. So it's the commercially most relevant species for cotton crops. The status of the genome sequences of this species is as follows. So the diploid donor species of Gossypium hirsutum have been sequenced. So the A genome species, G. arboreum, and the D genome species have been sequenced and published before. And actually KeyGene engaged in draft sequencing of the tetraploid hirsutum species. Actually we produced 38x coverage de novo PAG Bio smart cell sequencing, and actually we also built this 10x back library coverage whole genome profiling physical map that we use for super scaffolding. I will come back to that later. But now, to start off with the isoseq part. Of course, isoseq is a very interesting and powerful long-read transcription sequencing technology, and especially for polyploid crops, or tetraploid crops in our case. We were interested in investigating to what extent we can also separate transcripts originating from the ancestral diploid species that contribute to these polyploid crops. So that's why we engaged in this collaboration with Pacific Biosciences, and we followed the procedures that are developed by Pacific Biosciences. I will not go through all the steps, but some of my predecessors here in this workshop have explained this process, RNA isolation, sequencing of different size fractions of transcripts with the PAG-BioRS platform, and then it goes through the bioinformatics pipeline to identify unigenes and isoforms. So the experimental design that we took was the following. We obtained we started with actually a commercially available cultivar MRC 5156, an isolated RNA from 3 tissues, leaf, stem and root. So we took one homogenized sample of each tissue. From these tissues, 4 library types were constructed. This was actually done by Pacific Biosciences and sized according to the different size fractions shown on this slide. And then from each library of each tissue and each size selection, 4 SMART cells were sequenced, totaling 48 SMART cells in total. So the results were that between 16,000 and 19,000 unigenes were extracted from this data set, the highest number in leaf. The number of unique transcripts ranged from 19,500 to almost 25,000, again with the highest number in leaf, and between 38,000 and 46,795 high quality isoforms were extracted from these 48 SMART cells. So it's really a enormous data set that we are just at the very beginning of understanding what it all means. But the initial analysis that we did was, of course, well, can we identify transcripts that are specific to the A or the D genome? And this slide shows an example of such a transcript, which not only has 2 haplotypes, as you can see if you compare the top to the bottom, where in the bottom, the A genome transcript, there are 3 SMP polymorphisms that are absent from the transcripts in the D genome, but both actually also contain a splice variant, which is actually common to the DNA genome. So in total, actually, we have 4 different haplotypes identified in this transcriptome sequencing data set. Another interesting example is a clear example of exon skipping. The sequence shown in red is actually a complete exon that is differentially spliced in leaf tissue, as is also shown on this easier to understand depiction of the gene organization of this gene, where you can see that the small exon, this the second from the left, is actually missing in this alternatively spliced transcript. So at this point already we can conclude that iso-seq sequencing is a very powerful tool and a very informative tool to disentangle transcripts originating from ancestral genomes, as well as to identify splice variants and exon skipping events. So I will now proceed with explaining what we did in terms of de novo genome sequencing. This was done actually with an other cultivar, but belonging to the same Gossypium hirsutum species. Our aim was to generate approximately 40x coverage initially, using 20 kb sized libraries, using the blue pippin approach. And we sequenced a total of 242 SMART cells. And these data were processed and assembled using Falcon. And also there we collaborated with Pacific Biosciences. This assembly was actually done in the Amazon cloud and took 3 days to complete. Actually, I still recall that shortly before Christmas we had a conference call to align how we got Falcon running in the cloud, and by Monday morning when I got back in the office I had the results on my desk. So I was really impressed with the speed at which this tool can work if you have sufficient computer power behind that. So that's really amazing. So these are the results of this assembly. Again, it was 38 coverage in total. yielding in excess of 21,000 contigs with an average, with an N50 contig length of 218 kb. But importantly also, we were very impressed with the N90 specs, namely an N90 of almost 50 kb and an N90 index of almost 10 kb. So, but these, of course, are metrics. The next question is, well, what's the quality of this assembly? So we did three different approaches to get an impression about the accuracy of this assembly. First of all, we blasted two public Unigene databases against the Falcon assembly. And secondly, we used the whole genome profiling physical map to determine which fraction of whole genome profiling tags actually map to the Falcon assembly, and I will come back to that, how that worked, to get an estimate of the combined effect of base accuracy and coverage. And we deliberately searched for possible misassemblies based on using the sequenced A and D genomes in combination with annotation of these physical map contigs, and I will come back to that as well. So to start off with the Unigene analysis, I mentioned we took two independent Unigene data sets, and actually the results of this comparison indicated that between 73 and 79% of the Unigenes matched at over 90% of the total length to the Falcon contigs, which is actually pretty good number. And we've earlier generated a de novo physical map of this same accession with about 300x coverage Illumina, assembled with platanus, and actually did the same validation, and actually the percentages were virtually identical. So that was the first indication that in terms of coverage, these assemblies are likely to be comparable. So whether they comprise exactly the same fraction, we don't know, but the percentages of the Unigenes are virtually identical, within tenths of a percentage. So next we used the whole genome profiling map in this quality assessment. I will briefly explain how whole genome profiling works. Actually, it's our back-based physical mapping technology that we developed in the past. It's based on individual backclones that are arrayed in usually 384-well plates that are pooled. So, for example, row and column pools are made, and from these pooled backs, actually restriction enzyme digests are made in order to create sequence tags that are basically skim sequencing these backclones. But since restriction enzymes are used, identical tags can be obtained from backs that comprise the same region of the genome. And actually these sequence tags are then assigned to the individual backs, knowing the pooling scheme. And based on these tags, physical maps are built of overlapping backclones. And that's actually yielding a sequence-based physical map. And since it is sequence-based, it can be immediately integrated with a de novo genome sequence. So we have this physical map available for the same cotton line. And actually, 57% of the whole genome profiling tags uniquely match to a single whole genome sequencing contig. That's a pretty good number, but I would like to have seen it a little bit higher. What you see at the bottom is that there were 37% of the whole genome profiling tags which did not have a match. And at this point in time, we don't really know what is the explanation for that. At this moment, I believe that possibly the depth of sequencing that we did with the Pacific Biosciences sequencing approach of 38x is at the lower side for a tetraploid crop. And with some deeper sequencing, we would probably have a lower percentage of tags that have no match to Falcon contigs. But the truth is that we do not really know at this point in time. So the final quality control step that we did is making use of the published A and D genome sequences. Actually, we took 1,467 back contigs that we annotated with unique whole genome profiling tags unique to the A or the D genome. So we basically split up these 1,467 back contigs in 825 that were clearly derived from the A genome and 414 that were clearly derived from the D genome. And then we identified Falcon contigs which actually were matching more than one whole genome profiling contig to see whether, in case multiple contigs were aligning with this Falcon contig, whether they were both or all derived from the A or the D genome, as that is what you would expect for a high quality assembly. So while this is not a comprehensive analysis to validate the entire map, at least it gives insight into the consistency between the sequence-based physical map and the Falcon contig assembly. So to the extent that we could do that, there was actually very little indication of contigs comprising both A and D whole genome profiling contigs. 89 comprised A and D genome sequence, so that's 0.4% of the total number of contigs, which actually could be ancestral recombination breakpoints or misassemblies. We do not know. The fact that they cover both is not necessarily indicating that anything is wrong. It's just an observation. Whereas there were 22 examples, which represents 0.1% of the total of 21,000 Falcon contigs, where we believe that there is a collapsed assembly and therefore, let's say, a misassembly. But in conclusion, we have we believe there's very little indication of misassemblies, and we believe that these annotated contigs are actually very useful to validate assembly accuracy and also to identify ancestral recombination breakpoints in polyploid organisms. And this is something we actually have done in the past in tobacco, which also is an allotetraploid, and this actually is data that we've published 2 years ago, I believe, together with Philip Morris, and was presented here at PAG as well. So now onto the scaffolding part of the de novo assembly. I make a small sidestep to melon. Like many of the people here, we have in the past used other platforms. We started out with melon sequencing at the 454 platform 8 years ago. We have last year did a de novo genome sequencing effort with PacBio sequencing alone for a total of 44x coverage, and well, you can see from the metrics that of course the number of contigs go down. The N50 dramatically goes up from 12.8 kb to 387 kb. But the question that we wanted to address is if you integrate such a de novo genome sequence generated with Pacific Biosciences with a whole genome profiling map, at what depth of coverage of this physical map is necessary to obtain a certain superscaffolding efficiency. So also for melon we had a whole genome profiling map comprising 12x back coverage in total, and we did what we call a decremental analysis. So we took subsets of the initial backs of 6x, 8x or 10x coverage, as well as the total of 12x. We built the physical maps from that, and you can see, of course, that translates directly to the number of backs that were included in this project. It was 18,800 at 6x and over 34,000 backloads at 12x. And the number of contigs, so these are back contigs, of course, decrease with increasing back coverage. So at 6x you get 534 contigs, back contigs, and at 12x 399. I realize this is not a lot of numbers, but what we were really interested in is, well, okay, if you have these physical maps at various depths of coverage, if you integrate that with the PacBio sequence assembly, how many superscaffolds do you get? So for melon, we had 6,332 PacBio contigs, and after integration with 6 to 12x sequence-based physical map contigs, the number of superscaffolds ranged between 206 for the 12x and 271 for the 6x. So, put it differently, already at 6x back library coverage you get a 23-fold less number of superscaffolds than you have contigs. So this really illustrates the complementarity between PacBio sequencing for de novo sequence assembly and whole genome profiling, back-based physical map construction for superscaffolding. And accordingly, the N50 sizes of 387K for the contigs improved to in excess of 2 million base pairs for the superscaffolds. So this was a sidestep in melon. Of course, this talk is about cotton. So last week, actually, we integrated our PAG Bio de novo assembly, shown here at the right-hand side, 21,777 contigs, with a 10x back library coverage sequence-based physical map. And we actually got comparable results. After integration, we had 1,200 superscaffolds. And you see the N50 values. The N50 value of the superscaffolds is 2.3 million bases, where we came from 280 kb N50 for the contigs. And similar metrics for the N90 and the N90 index. So this is a second example where we show that PAG Bio de novo sequencing and whole genome profiling sequence-based physical mapping actually reinforce each other, both in terms of superscaffolding as well as using it for quality control purposes. So I will be reaching the end of my presentation. Any genome assembly will have remaining gaps, we all know that. And in addition to that, the breeders or the molecular breeders that we work with often have an interest in specific regions in such genome assemblies, because that's where their genes of interest are located. So we developed a technology which we call a targeted gap filling or a targeted resequencing approach to fill remaining gaps in the assembly. It's actually based on, again, a restriction enzyme mediated approach. So we take advantage of the restriction enzyme recognition sequences that are closest to the gaps that we want to fill, and we construct a partial digest of the genome of this organism, and then ligate adapters to this partial digest, circularize the molecules, and then perform a whole genome amplification on these circular molecules, and then subject this whole genome amplification product to Pacific Biosciences sequencing. So whereas there are limitations in long amplicon sequencing by PCR, this method is not PCR based, and actually this comes in where PCR ends. That's how we like to phrase it in our company. So we used maize as a model to develop this technology. The maize genome, actually, it was mentioned earlier, I think, in this presentation, contains still a large number of gaps. In many instances, actually, the gap size is not specifically known, and they are arbitrarily set at 100 base pairs. But what we did is we took a 1 million base pair region of the chromosome 4 of maize, containing 64 gaps, and applied this method. And in the first attempt, actually, the results are shown at the bottom. We filled 25 of them, either closed them completely or reduced their size. 18 were overfilled, 5 were extended, and 16 in 16 cases this method failed. So we are actually quite pleased with that. We are using this technology now to close gaps in regions of interest. So I mentioned the Cottonpedia in the beginning. This approach is all about integration. We have a de novo genome sequence assembly. We have transcriptome sequences of various tissues. And of course, being in a breeding research company, we have many more genetic maps and QTLs and other relevant literature information in cotton. And we put it all together in this genome encyclopedia called Cropedia, which actually integrates both information in the public domain and proprietary data by our clients. And actually it comes with a web-based interface for easy navigation. And as of now, we have actually made Cropedias for 16 different crops. And this is actually the dashboard entry to this Cropedia, where you can select which type of data you want to access, and it's all interlinked. So all the isoseq data of cotton will be integrated in this Cottonpedia as well. So to wrap up our track record, we have been happily using the Pacific Biosciences platform for a couple of years now. We have sequenced a large number of plant organelles, mitochondrial and chloroplast genomes, more than 5,000 bags, more than 250 microbial genomes, plant pathogen genomes, crop genomes. I explained some of that here. We've done several full-length transcriptome projects, including isoseq, and long amplicon targeted sequencing or resequencing projects, for example, for mutation validation. And we were very happy by the quote by Mike Hunkerpiller last year in the Sequence that with these efforts we have developed ourselves into one of the highest capacity PAG-Bio users in Europe, and we hope it will stay that way. In summary, we have applied an integrated transcriptome and genome sequencing approach in cotton to create a high-quality genome catalog, including tissue-specific unigenes and isoform gene sets. Identification, we showed identification of A and D genome-specific transcripts and splice variants, and a genome assembly with an N50 value of more than 200 kb and less than 1% misassembly. We believe the whole genome profiling technology and PAG-Bio de novo genome sequencing are highly complementary, as shown by the 5 to 10-fold higher N50 superscaffold size than the context size and the 20 to 30-fold less number of superscaffolds than context. We have a targeted resequencing technology available to close gaps, and we integrate all these resources in Acropedia. So I would like to thank a large number of people, actually. I will not go through all of them, but some of them I have to certainly mention. First of all, Yifeng Tang at KeyGene, who collaborated intensively with Elizabeth Steng at Pacific Biosciences to analyze the isoseq transcriptome data. Similarly, Mike Cariasso and Tyson Clark, who were instrumental in the Falcon de novo assembly. Tin Hong for making the cDNA libraries. A number of other people, and Dwakesh Parihar for supplying the tissues for, or actually the plants for the isoseq sequencing project. And with that, I would like to thank you for your attention.
Questions?
When you were talking about the melon PAG-Bio assembly and the backs, when you introduced the between 6 to 12x back coverage, the largest, I believe, super scaffold, I think is what you called it, increased and then decreased. Is there a reason for that, or was that a misassembly possibly, or?
I have to actually go back. I don't think there is a particular reason for that.
I think
Are you
one
referring
more
to this
forward.
slide?
Yeah, so the largest size, you have 7.8 to 10 and then 9 and 7 again.
Yeah, actually the largest is at 8x. I don't have a particular, it's about the largest size. I don't have any particular explanation for that. I think it probably relates to the way that the contigs are actually built, so the mathematics behind the assembly algorithm. But I don't think it's hugely disturbing. No.
Okay. Did you, by any chance, check them or
no?
Or no?
No, I
okay.
have not checked them. No.
Other questions?
If you increase the PAG-Bio X coverage, can you get away without doing the whole WGP?
Yeah, that's a very good question, and well, actually, what I believe is that with a higher depth of coverage of the PAG-Bio sequencing, we will probably get better N50 metrics than what we have achieved in the case in cotton. But I would certainly very much doubt whether there would be a 20 to 30-fold lower number of contigs. So the number of contigs will go down, for sure, but not at this rate. And it's not so strange, because a single backclone alone, already, before making a sequence-based physical map, is between 125 and 150 kb. So the scaffolding power of a back-based physical map is really tremendous. Yeah, but it's finding the right balance. That's the
So what about
behind it.
This technique? Have you applied for the high-seq or the other assembly?
Yes, in the past we have used sequence-based physical mapping, whole genome profiling maps to integrate with Illumina assemblies. And given the fact that the N50 contigs are so much smaller, the improvement in combination with whole genome profiling is of course very much larger, and that's why we ask ourselves the question, well, how does that work in case of Pacific Biosciences sequences, which clearly deliver much larger contigs, but even then there is still very strong complementarity.
Thanks.
Other questions? Okay, with that, I'd like to thank again each of our speakers today for all their time and efforts here this afternoon. Thank you.